Most of you will know that females have XX chromosomes and males have XY chromosomes. But have you ever wondered why females' bodies are able to only just encode genes for one X chromosome and not the other? In other words, why does the female body not duplicate every gene on the X chromosome twice? Well, the reason every cell in the female body doesn't duplicate all of the genes on every X chromosome is because of the Lion hypothesis. The Lion hypothesis states that even though there are two X chromosomes in every somatic female cell, one of them will be deactivated to prevent duplication of genes and traits expressed throughout the body. A good analogy for the Lion hypothesis is this umbrella I have with me. You can see it's fully extended, and if I went outside in the rain right now, it would prevent rain from falling on me. But what if I wanted to go inside and I didn't need it to be used to catch rain? Well, I could tightly fold it up, and then it would be much more convenient in my apartment. Well, this happens in female somatic cells also. The unused X chromosome is tightly compacted into what's called a bar body, and then it won't be used to duplicate information that's already present. A visible example of the Lion hypothesis in nature would be in the tortoiseshell cat. So some female cats are what's called tortoiseshell. The reason this occurs is because the gene that encodes for fur color is present on the X chromosome. So females might get one gene that encodes for black fur and another X chromosome that encodes for yellow fur. And because the somatic cells randomly deactivate one X chromosome throughout the body, you can have random splotches of black fur and yellow fur that appear all over the body of some cats. The fact that the tortoiseshell pattern is expressed randomly is important in the topic of the Lion hypothesis. So two clones of the same genetics would have a completely different tortoiseshell pattern across their body because the X chromosome that is deactivated is completely random throughout the body. Here I have an example written out of when a yellow short-haired female with this genotype, X little b, and then the HH alleles for hair, is crossed with a black male who's heterozygous for long hair. So X big B and a Y indicating it's a male, and then the capital H little h genotype for heterozygous long hair. As you can see here, the possible genotypes, you could be black, yellow, tortoiseshell, you could have long hair, or short hair. So here are the crosses, and you can see the Punnett squares I drew at the top. Well, one half of them, of the progeny from this cross, would be tortoiseshell, and the other half would be yellow. All of the females would of course be tortoiseshell, and all of the males would be yellow. Well, out of these half that are tortoiseshell, one half will have long hair and one half will have short hair. And in the one half that are born yellow, half of them will have long hair and half of them will also have short hair. You can see the total phenotypes here on the right. 1/4 of them will be tortoiseshell with long hair, 1/4 of them will be tortoiseshell with short hair, 1/4 of them will be yellow with long hair, and 1/4 will be yellow with short hair.